Assalamualaikum, I am Sana Kassam from batch 8 and today my presentation topic is algal bioreactors for carbon dioxide capture. First of all, we will discuss the introduction of the topic. The continuous increase in atmospheric carbon dioxide concentration due to industrialization, fossil fuel combustion and anthropogenic activities has become a major global environmental concern. Elevated carbon dioxide levels are directly linked to climate change, global warming, ocean acidification, and ecological imbalance. Conventional carbon capture and storage technologies such as chemical absorption, adsorption, geological sequestration, although effective, are often associated with high energy consumption, operational costs, and environmental risks. In this context, algal bioreactors have emerged as a sustainable and biologically driven alternative for carbon dioxide capture and utilization. Algal bioreactors are engineered systems designed to cultivate microalgae or cyanobacteria under controlled conditions to enhance their natural ability to assimilate carbon dioxide through photosynthesis. Microalgae utilize carbon dioxide as a carbon source and convert it into biomass in the presence of light, water, nutrients, etc. Unlike terrestrial plants, microalgae exhibit significantly higher photosynthetic efficiency, faster growth rates, and greater carbon dioxide fixation capacity, making them highly suitable for large-scale carbon dioxide capture applications. The fundamental principle of algal bioreactors for carbon dioxide capture lies in the biological conversion of inorganic carbon into organic matter. During photosynthesis, microalgae absorb carbon dioxide and using solar energy convert it into carbohydrates, lipids, proteins, and other valuable biochemical compounds. This process not only reduces carbon dioxide emission but also generates algal biomass that can be further utilized for the production of biofuels, bioplastics, animal feed, fertilizers, pharmaceuticals, and other high-value products. Therefore, algal carbon dioxide capture represents a carbon capture and utilization approach rather than simple storage. Algal bioreactors can be integrated with industrial emission sources such as power plants, cement factories, steel industries, where flue gas rich in carbon dioxide is directly supplied to the bioreactor. The integration enables the simultaneous mitigation of greenhouse gas emissions and the production of renewable biomass. Compared to open pond systems, closed photobioreactors often better control over environmental parameters such as light intensity, temperature, pH, nutrient concentration, and gas exchange, resulting in improved carbon dioxide uptake, efficiency, and biomass productivity. Next, we will discuss the composition and construction materials for algal bioreactors. Algal bioreactors, particularly photobioreactors used for carbon dioxide capture, are engineered systems composed of biological, structural, and functional components. This selection of materials is critical to ensure optical transparency, chemical stability, mechanical strength, biocompatibility, and long-term operational durability. First part is reactor vessel material. First we will discuss its first component. The reactor vessel is the main structural component that holds the algal culture and facilitates light penetration and gas exchange. It is further composed of glass, which is glass borosilicate glass, which is commonly used in laboratory scale algal bioreactors due to its excellent optical clarity, chemical inertness, and a resistance to corrosion. Glass does not react with nutrients or metabolic byproducts and allow uniform light transmission, which is essential for effective photosynthesis. However, its high cost and fragility limits its use at industrial scale. Next part is plastic polymers. Various transparent polymers are widely used for pilot and industrial scale bioreactors. Polymethyl, polycarbonate, polyethylene, polyvinyl chloride. These materials offer good light transmission, lower cost, flexibility in design, and resistance to environmental degradation. And the next one is tubing and piping materials. Tubular photobioreactors consist of long transparent tubes that maximize the surface area to volume ratio. Transparent polyethylene or polycarbonate tubing is used to allow maximum light penetration. Silicone or rubber tubing is employed for gas transfer due to its flexibility and permeability. These materials must be resistant to biofouling pressure variations and prolonged exposure to sunlight. And the next part is gas exchange and sparging components. Effective carbon dioxide delivery is achieved through gas sparging systems. Stainless steel spargers are used due to their corrosion resistance and mechanical strength. Ceramic or porous polymer diffusers generate fine glass bubbles to enhance carbon dioxide dissolution. All gas contact materials must tolerate acidic conditions caused by dissolved carbon dioxide. The next is mixing and circulation components. Proper mixing prevents algal sedimentation and ensures uniform exposure to light and nutrients. Mechanical stirrers and impellers are typically made of stainless steel or chemically resistant polymers. Airlift systems rely on gas flow rather than mechanical parts, reducing material wear and contamination risk. Pump components are often constructed from polypropylene or Teflon. Next is light delivery systems. Light availability directly affects reactor efficiency. Transparent reactor walls serve as the primary medium for natural light penetration. LED light panels are used in indoor systems or for controlled illumination. Reflective materials such as aluminum coated surfaces are sometimes installed to enhance light distribution. Next is nutrient medium composition. Although not structural materials, the cultural medium is a critical internal component. Its further parts are water, nitrogen sources, phosphorus sources, trace metals, buffering agents to maintain pH stability. The medium must be compatible with the reactor materials to prevent leaching and degradation. The next is sensors and control materials. Modern algal bioreactors include integrated monitoring systems, pH electrodes, temperature sensors, and dissolved oxygen probes housed in chemically resistant cases. Materials used include glass electrodes, polymer housings, and stainless steel connectors. The next part is harvesting and separation components. Biomass recovery units are constructed using centrifuge bowls made of stainless steel, filtration membranes composed of polymeric materials, flocculation tanks lined with corrosion-resistant coatings. Next, we will discuss the working mechanism of algal bioreactors for carbon dioxide capture. We will see how they will work. The working mechanism of algal bioreactors for carbon dioxide capture is based on the biological fixation of carbon dioxide through photosynthesis under controlled reactor conditions. This process integrates principles of chemistry, biochemistry, mass transfer, reactor engineering to maximize carbon uptake and biomass productivity. Carbon dioxide supply and gas transfer. Carbon dioxide is supplied to the algal bioreactor either from ambient air or directly from industrial flue gases containing high carbon dioxide concentrations. The gas is introduced into the reactor through spargers or gas diffusers, producing fine bubbles that enhance gas-liquid mass transfer. Dissolved carbon dioxide in the aqueous medium exists in equilibrium with bicarbonate and carbonate species depending on the pH of the culture medium. Efficient dissolution of carbon dioxide is essential as it directly influences the availability of inorganic carbon for algal uptake. The next is light absorption and photosynthetic activation. Light is a crucial energy source for algal metabolism. In photobioreactors, light is supplied either by natural sunlight or artificial illumination and distributed uniformly to avoid photoinhibition or shading effects. Algal cells contain photosynthetic pigments such as chlorophyll A, chlorophyll B, and carotenoids, which absorb light energy and initiate the light-dependent reactions of photosynthesis. These reactions generate adenosine triphosphate and reduced nicotinamide adenine dinucleotide phosphate, which are required for carbon fixation. And the next part is carbon fixation via the Calvin cycle. Once carbon dioxide is dissolved in the culture medium, it is transported into algal cells through diffused or specialized carbon concentrating mechanisms. Inside the chloroplast, carbon dioxide is fixed through the Calvin-Benson cycle. The enzyme ribulose 1,5-biphosphate carboxylase or oxygenase, which is also called Rubisco, catalyzes the carboxylation of ribulose 1,5-biphosphate, leading to the formation of organic compounds. This biochemical step is the core mechanism through which inorganic carbon is converted into biomass. Next is biomass growth and carbon dioxide assimilation. The fixed carbon is subsequently used for cellular growth and metabolism, resulting in the synthesis of carbohydrates, lipids, proteins, and nucleic acids. As algal cells multiply, a continuous removal of carbon dioxide from the system occurs. The rate of carbon dioxide capture depends on factors such as algal species, light intensity, nutrient availability, temperature, and pH. Controlled reactor conditions ensure optimal growth and sustained carbon sequestration. Next is oxygen release and pH regulation. During photosynthesis, oxygen is released as a byproduct. Accumulation of oxygen can inhibit algal growth and reduce carbon dioxide solubility. Therefore, proper gas exchange is maintained to remove excess oxygen. Additionally, carbon dioxide uptake causes an increase in pH due to the consumption of dissolved carbon species. pH control systems are often employed to maintain optimal chemical conditions for algal metabolism and to enhance carbon dioxide absorption efficiency. Next is biomass harvesting and carbon utilization. After sufficient biomass accumulation, algal cells are harvested using methods such as centrifugation, filtration, flocculation, or sedimentation. The harvested biomass represents captured and biologically stored carbon, which can be further processed into biofuels, biochemicals, or other value-added products. This step transforms carbon dioxide capture into a carbon utilization pathway, contributing to a circular carbon economy. The next one is continuous or semi-continuous operation. Modern algal bioreactors often operate in continuous or semi-continuous modes where fresh nutrients and carbon dioxide are supplied while biomass is periodically removed. This operational strategy maintains steady-state conditions, maximize productivity, and ensure long-term carbon dioxide capture efficiency. Carbon concentrating mechanisms. Carbon concentrating mechanisms are physiologically adaptations that increase carbon dioxide availability around Rubisco. These mechanisms involve active transport of bicarbonate ions and their conversion to carbon dioxide within specialized cellular compartments. Next, we will discuss the types of algal bioreactors that is used to capture carbon dioxide. First type is open pond system. Open pond systems are the simplest and most traditional form of algal cultivation. Description of this type is these systems consist of shallow ponds and raceway channels where algae are grown under natural sunlight and atmospheric conditions. Carbon dioxide is supplied either from air or injected from industrial exhaust gases. Its characteristics are large surface area exposed to sunlight, typically mixed using paddle wheels, low construction and operating cost. Its advantages are economical and easy to construct, suitable for large-scale biomass production. It also has some limitations, which are low carbon dioxide utilization efficiency, high risk of contamination, limited control over temperature, pH, and light. Due to these limitations, open ponds are less preferred for efficient carbon dioxide capture applications. The second type is closed photobioreactors. Closed photobioreactors are scale systems that provide controlled growth conditions, making them highly efficient for carbon dioxide capture. Tubular photobioreactors. Tubular photobioreactors consist of long transparent tubes arranged horizontally, vertically, or in helical configurations. Carbon dioxide is injected into the circulating algal culture. Its advantages are high surface area to volume ratio, enhanced carbon dioxide mass transfer, reduced contamination risk. It also has some limitations, and these limitations are oxygen accumulation can inhibit growth, high construction and maintenance cost. Tubular are widely used in industrial scale carbon dioxide capture projects. The third type is flat panel photobioreactors. Flat panel reactors are made of flat transparent plates that allow uniform light penetration across the culture. Its advantages are efficient light utilization, high biomass productivity, easy temperature control. And its limitations are limited scalability, higher material cost. These reactors are vertically oriented systems and are classified into two main types. Bubble column reactors. Carbon dioxide is introduced at the bottom, creating gas bubbles that provide mixing and mass transfer. Its advantages are simple design, low energy consumption, good gas liquid contact, and its limitation is limited light penetration in large columns. Another is airlift photobioreactors. These systems use a riser and downcomer to create circulation using air or carbon dioxide. Its advantages are improved mixing efficiency, lower shear stress on algal cells, better carbon dioxide absorption. Airlift reactors are suitable for delicate microalgal species. Third type is hybrid algal bioreactors. Hybrid system combine open ponds with closed photobioreactors. Algae are initially grown in closed reactors for high purity and then transferred to open systems for mass cultivation. Its advantages are balanced cost and productivity, improved contamination control, and its limitation is complex operational management. Fourth type is membrane photobioreactors. These advanced systems use gas permeable membranes for controlled carbon dioxide diffusion into the algal culture. Its advantages are high carbon dioxide transfer efficiency, precise control over gas exchange, and its limitations are high capital cost, membrane fouling issues. Fifth type is biofilm based algal reactors. In these systems, algae grow as biofilm on solid surfaces rather than in suspension. Its advantages are reduced harvesting cost, high carbon dioxide fixation rates, and its limitation is limited large-scale implementation. Now we will discuss the properties of algal bioreactors for carbon dioxide capture. Algal bioreactors exhibit a combination of biological, chemical, physical, and operational properties that make them effective systems for carbon dioxide capture and utilization. These properties determine their efficiency, stability, and suitability for large-scale environmental applications. The first property is high carbon dioxide fixation efficiency. One of the most important properties of algal bioreactor is their ability to efficiently capture and assimilate carbon dioxide. Microalgae possess high photosynthetic rates and can utilize carbon dioxide concentrations significantly higher than atmospheric levels. Under optimized conditions, algal systems can capture carbon dioxide more rapidly than terrestrial plants. The second property is photosynthetic activity. Algal bioreactors rely on oxygenic photosynthesis where light energy is converted into chemical energy. The presence of photosynthetic pigments such as chlorophyll and carotenoids enables effective absorption of light, driving carbon fixation through enzymatic pathways. The third property is high biomass productivity. Algal bioreactors are characterized by rapid algal growth and high biomass yield. Microalgae can double their biomass within hours or days, resulting in continuous carbon assimilation and efficient utilization of reactor volume. Fourth property is light utilization efficiency. Efficient light distribution is a key property of algal bioreactors. Transparent reactor materials and optimized geometry ensure uniform light penetration, minimizing shading effects and maximizing photosynthetic efficiency. Fifth property is gas liquid mass transfer capability. Algal bioreactors are designed to enhance carbon dioxide dissolution and transfer from the gas phase to liquid culture medium. Fine gas bubbles, optimized mixing, and controlled flow rates improve carbon availability for algal uptake. Sixth property is pH stability and buffering capacity. Carbon dioxide absorption and photosynthetic influence the pH of the culture medium. Algal bioreactors exhibit controlled pH conditions through buffering systems or regulated carbon dioxide input, maintaining an optimal chemical environment for algal metabolism. Seventh characteristic is temperature tolerance and control. Algal growth occurs within specific temperature ranges, typically between 20 to 35 degrees Celsius. 20 to 35 degrees Celsius depending on species. Bioreactors allow temperature regulation, ensuring stable metabolic activity and consistent carbon dioxide capture rates. Its eighth characteristic or property is nutrient utilization efficiency. Algal bioreactors efficiently utilize inorganic nutrients such as nitrogen, phosphorus, and trace metals. This property allows integration with wastewater streams where nutrients are recycled while simultaneously capturing carbon dioxide. Next is oxygen evolution and management. Oxygen is produced as a byproduct of photosynthesis. Effective algal bioreactors are designed to remove excess dissolved oxygen, preventing oxidative stress and inhibition of carbon dioxide uptake. The next is scalability and operational flexibility. Algal bioreactors possess scalable designs ranging from laboratory to industrial scale. They can operate in batch, semi-continuous, or continuous modes, offering flexibility for different carbon dioxide capture applications. The next is carbon capture and utilization capability. A distinct property of algal bioreactor is their ability to convert captured carbon dioxide into valuable biomass. The produced biomass can be further processed into biofuels, biochemicals, animal feed, fertilizers, enhancing their economic feasibility of carbon dioxide mitigation. And the last one is environmental sustainability. Algal bioreactors are considered environmental friendly systems due to low secondary pollution, renewable biomass generation, reduced carbon footprints. Now we will see the chemistry of algal bioreactors for carbon dioxide capture. The chemistry of algal bioreactors is governed by a combination of aqueous carbon chemistry, photosynthetic biochemistry, enzyme catalyzed reactions, and cellular metabolism. These chemical processes collectively enable the conversion of inorganic carbon into stable organic biomass. First is aqueous carbon dioxide chemistry and speciation. When carbon dioxide is introduced into the algal bioreactor, it dissolves in the aqueous culture medium and establishes a dynamic chemical equilibrium. Dissolved carbon dioxide reacts with water to form carbonic acid. Carbonic acid partially dissociates into bicarbonate and carbonate ions. The relative concentration of carbon dioxide, bicarbonate, carbonate depends strongly on pH. In algal cultures, bicarbonate is often the dominant inorganic carbon species and serve as a major carbon source for many microalgae. The second is carbon concentrating mechanism. Many algal species possess carbon concentrating mechanisms that actively transport bicarbonate and carbon dioxide into the cell. These mechanisms increase the intracellular carbon dioxide concentration near the active site of the enzyme ribosco, overcoming its low affinity for carbon dioxide. Key chemical components that is used in carbon concentrating mechanisms are bicarbonate transporters, carbonic anhydrase enzyme. Carbonic anhydrase catalyzes the rapid interconversion between carbon dioxide and bicarbonate. And the third is photosynthetic light reactions, which is also called photochemistry. The light-dependent reactions of photosynthesis occur in the thylakoid membrane and involve photochemical processes. Key reactions include absorption of photons by chlorophyll, water oxidation at photosystem 2, electron transport leading to the formation of ATP and NADPH. These reactions provide the chemical energy required for carbon fixation. The next is carbon fixation via the Kelvin-Benson cycle. The core chemical pathway for carbon dioxide capture is the Kelvin cycle, which consists of three main stages: carboxylation, reduction, regeneration. And the next is biomolecular synthesis and carbon storage. In this, the fixed carbon is chemically incorporated into carbohydrates, lipids, proteins, amino acids, etc. And the next is pH chemistry and buffering effects. As carbon dioxide is consumed during photosynthesis, the concentration of hydrogen ions decreases, leading to an increase in pH. Chemical buffering systems are used to stabilize the pH and maintain optimal reaction conditions. And then oxygen chemistry and oxidative balance. Oxygen produced during photosynthesis can participate in side reactions including photorespiration via ribosco oxygenase activity, formation of reactive oxygen species. Now we will discuss the linkage of algal bioreactors for carbon dioxide capture with Sustainable Development Goal SDGs. First of all, it is linked with SDGs 13 climate action. Algal bioreactor directly address SDG 13 climate action by reducing atmospheric and industrial carbon dioxide emission through photosynthetic carbon fixation. Microalgae convert carbon dioxide into organic biomass, thereby mitigating greenhouse gas accumulation and slowing global warming. Unlike conventional carbon capture and storage, algal systems offer carbon capture and utilization, ensuring long-term environmental benefits. Then SDG 7 affordable and clean energy. Algal biomass produced during carbon dioxide capture can be converted into biofuels such as biodiesel, bioethanol, and biogas. This supports SDG 7 by promoting renewable and low carbon energy sources. The dual function of carbon dioxide mitigation and bioenergy generation enhances energy sustainability and reduces dependence on fossil fuels. The next one is SDG 9 industry innovation and infrastructure. The development and scaling of algal bioreactors encourage technological innovation and sustainable industrial infrastructure. Supporting SDG 9 integration of algal bioreactors with power plants, cement factories, steel industries enable cleaner production systems and promotes environmentally responsible industrial growth. The next is SDG 12 responsible consumption and production. Algal bioreactors contribute to SDG 12 by transforming waste carbon dioxide into valuable products such as biofertilizers, animal feed, pigments, and bioplastics. This approach supports resource efficiency, waste valorization, and sustainable product cycles, key principles of responsible consumption. The next one is SDG 6 Clean Water and Sanitation. Many algal bioreactor systems can be coupled with wastewater treatment where algae utilize nutrients such as nitrogen and phosphorus. This contributes to SDG 6 by improving water quality, reducing nutrient pollution, and enabling water reuse while simultaneously capturing carbon dioxide. The next one is SDG 20 Hunger. Algal biomass is rich in proteins, vitamins, and essential nutrients when used as animal feed or biofertilizers. It supports agricultural productivity or food security, contributing indirectly to SDG 20 Hunger. Next is SDG 11 Sustainable Cities and Communities. By reducing industrial emissions and improving air quality, algal bioreactors help create cleaner urban and industrial environments. Supporting SDG 11, their integration into urban infrastructure promotes sustainable city development. The next is SDG 8 Decent Work and Economic Growth. The algal biotechnology sector generates green jobs in research, engineering, biomass processing, and renewable energy industries. This supports SDG 8 by fostering sustainable economic growth and employment opportunities. The next is SDG 14 and 15 Life Below Water and Life on Land. Reduction in carbon dioxide emission helps limit ocean acidification and protects terrestrial ecosystems. Additionally, algae-based systems reduce pressure on land resources compared to traditional crops, preserving biodiversity. Overall sustainability perspective, algal bioreactor represents a holistic sustainability solution by capturing greenhouse gases, producing renewable biomass, recycling nutrients, supporting circular economy principles. Now we will discuss future prospects of algal bioreactors for carbon dioxide capture. The future of algal bioreactors for CO2 capture is highly promising due to increasing global emphasis on climate change, mitigation, carbon neutrality, and sustainable industrial practices. Continuous advancements in biotechnology, material sciences, and process engineering are expected to significantly enhance the efficiency, scalability, and economic feasibility of algal based carbon capture systems. Genetic and metabolic engineering of microalgae. Future research will focus on genetically engineered algal strains with enhanced carbon dioxide fixation rates, improved tolerance to high carbon dioxide concentrations, and reduced photorespiration, modification of rubisco activity, optimization of carbon concentrating mechanism, and regulation of lipid and carbohydrate synthesis pathways will lead to higher biomass productivity and more efficient carbon sequestration. The second one is advanced photobioreactor design. Next generation photobioreactors will incorporate novel materials with improved light transmission, self-cleaning surfaces, enhanced durability. Smart reactor designs using computational fluid dynamics will optimize light distribution, gas transfer, and mixing, resulting in higher carbon dioxide capture efficiency with lower energy input. The third one is integration with industrial emission sources. In the future, algal bioreactors are expected to be directly integrated with power plants, cement industries, steel manufacturing units, and refiners. Utilizing unrated flue gas as a carbon source will reduce operational cost while enabling real-time carbon capture at emission points. The fourth one is coupling with wastewater treatment systems. The integration of algal bioreactors with municipal and industrial wastewater treatment will become increasingly common. Algae can utilize nitrogen and phosphorus from wastewater, simultaneously achieving carbon dioxide capture, nutrient removal, and biomass generation, making the process more sustainable and cost effective. The fifth one is artificial intelligence and process automation. Future algal bioreactors will employ artificial intelligence and machine learning algorithms to monitor and optimize growth conditions, real-time control of pH, light intensity, carbon dioxide flow, and nutrient supply will maximize productivity while minimizing operational energy requirements. And the sixth one is biofuel and bioproduct diversification. Advancement in downstream processing will allow algal biomass to be converted into a wider range of high-value bioproducts. Seventh one is hybrid carbon capture systems. Future carbon mitigation strategies may combine algal bioreactors with conventional carbon capture technologies such as chemical absorption or mineral carbonation. Such hybrid systems will enhance overall carbon dioxide removal efficiency while reducing the limitation of industrial technologies. Eighth one is large scale commercialization or policy support, and the ninth one is contribution to carbon neutral and carbon negative goals. In the long term, algal bioreactors have the potential to contribute to carbon neutral and carbon negative industrial systems where CO2 emissions are fully offset or exceeded by biological capture and utilization. Now we will discuss the limitation and challenges for algal bioreactors for carbon dioxide capture. The first limitation is high capital and operational cost. One of the major limitations of algal bioreactors, particularly closed photobioreactor, is their high initial investment cost. Transparent construction materials, advanced lighting systems, sensors, and control units significantly increase capital expenditure. In additional operational cost related to energy consumption for mixing, pumping, temperature control, and artificial illumination remains high. The second limitation is energy intensity of the process. Although algal systems are environmentally friendly, they can be energy intensive, especially when artificial light sources and mechanical mixing are used. If renewable energy is not employed, the overall carbon balance may be reduced, compromising the sustainability of the system. Third limitation is light limitation and photo inhibition. Efficient light distribution is difficult to maintain at large scale. Dense algal cultures often experience self-shading, while excessive light can cause photoinhibition, damaging photosynthetic pigments and reducing carbon dioxide fixation efficiency. Optimizing light penetration remains a significant engineering challenge. The fourth limitation is mass transfer limitation. The transfer of CO2 from the gas phase to the liquid culture medium can be limited by poor gas liquid mass transfer efficiency. High CO2 flow rates may lead to bubbles and carbon dioxide loss, while insufficient supply restricts algal growth. The fifth limitation is oxygen accumulation. Photosynthesis produces oxygen as a byproduct. Accumulation of dissolved oxygen can inhibit algal metabolism, reduce carbon dioxide solubility, and increase photorespiration through rubisco oxygenase activity. Efficient oxygen removal systems are required but add to system complexity. Sixth limitation is biological contamination and culture stability. Algal cultures are vulnerable to contamination by unwanted microorganisms, including bacteria, fungi, and competing algal species. In open systems, contamination is particularly difficult to control, leading to reduced productivity and culture collapse. Sensitivity to environmental conditions. Microalgae are sensitive to variations in temperature, pH, salinity, and nutrient availability. Maintaining optimal conditions under fluctuating outdoor environments or industrial settings poses significant operational challenges. Another limitation is nutrient requirements and resource demand, biomass harvesting and processing challenges, scalability, and land requirements. Limited carbon dioxide capture capacity per unit area, regulatory and economic uncertainty. Regulatory limited carbon dioxide capture capacity per unit area, how it will affect, how it will be considered as its limitation compared to geological carbon storage or chemical absorption system. Algal bioreactors currently exhibit lower carbon dioxide capture capacity per unit area, requiring large installation to achieve significant emission reductions. And the next one is regulatory and economic uncertainty. Lack of clear regulatory frameworks, carbon pricing mechanisms, and market incentives for algal based carbon capture technologies slows commercial adoption. Economic viability often depends on co product valorization rather than carbon dioxide capture alone. And the lastly, we will conclude this topic. Algal Conclusion. Algal bioreactors represent a promising and sustainable technology for mitigation atmospheric and industrial carbon dioxide emissions by leveraging the natural photosynthetic capabilities of microalgae. Through controlled cultivation, these systems efficiently convert inorganic carbon into valuable biomass, which can be further utilized for biofuels, bioproducts, nutrient rich feed, supporting a circular bioeconomy. Their effectiveness is underpinned by advanced reactor designs, efficient light utilization, carbon concentrating mechanisms, and integration with industrial emission sources and wastewater treatment. While challenges such as high capital and operational cost, energy intensity, high light limitations, contamination risk, and scalability issues remain, ongoing innovation in genetic engineering, reactor engineering, process automation, and hybrid systems are expected to enhance productivity, economic feasibility. Furthermore, algal bioreactors align with multiple United Nations Sustainable Development Goals, including climate action, clean energy, responsible production, and sustainable industry, highlighting their multifunctional roles in addressing global environmental and socio-economic challenges. In essence, algal bioreactors offer a biologically robust, environmentally friendly, and commercially viable pathway toward carbon mitigation, renewable resources generation, and sustainable development. This is all from my side. Thank you.